Good afternoon. My name is Ania Bula, and I am an agriculture sciences student working with Dr. Lucy Lee. Cell culture is the science of growing cells outside of a living organism in an in vitro environment. It is a tool with many applications, from toxicological research that can investigate impacts directly on different tissues without the use of live animals, to being used to grow and develop lab-grown meat as a part of cellular agriculture. Its usefulness is dependent on the development of different cell lines. These may be used to represent a particular species or taxon, to represent specific tissue types like gut or skin cells, or different habitats such as marine or freshwater environments. To this end, I attempted to initiate a primary culture and develop a cell line from a local species, the Northern Pikeminnow, or Ptychocheilus oregonensis. But why would I choose to develop a cell line from a fish that is so hated by fishermen? Well, of the 960 fish cell lines currently listed on Cellosaurus, the cell line database, none have been isolated from this particular species. This means that I would be contributing to broadening the diversity that is on offer. In addition to being a novel species, this cell line was isolated from the olfactory passages, which is a tissue that is not well represented among those available. One reason why Northern Pikeminnow are so hated is because of their tendency to predate on juvenile salmon. Throughout their range, there are many conservation programs that are actually aimed at reducing their population. Yet despite this, they continue to thrive in many of the same waterways where salmonids struggle. Having cell lines of this species can be used to compare the sensitivities of salmonids to others in their environment in response to harmful stimuli such as pollutants and pests. To initiate the primary culture, I took a sample from the olfactory passages of a wild-caught pikeminnow and transferred it to a cell culture flask along with the appropriate media. The cells migrated from the sample into the flask, and since then, they've been transferred or passaged multiple times, creating almost 10 new generations of cells, the first successful steps in developing a new cell line. Thank you.